Hi everyone, we're picking up where we left off on day 12, and we're trying to make sure we understand how a bacterial cell should efficiently respond to different scenarios regarding glucose and lactose. So we know that the most efficient carbon source for bacteria is to use glucose directly. So if both are present, there's no reason to use lactose and convert it into glucose. And so we're going to talk about how this happens by understanding the CAP system. So when glucose is low, we're going to see high levels of cyclic AMP. And when there's high levels of cyclic AMP, that means that CAP will be bound by cyclic AMP, and this complex here, this blue CAP protein bound by cyclic AMP, will bind near the promoter, recruiting RNA polymerase to the promoter, and so we're going to get higher levels of transcription. And this makes sense. We want to be able to turn the system on at high levels if glucose is at low levels. In contrast, when glucose is at high levels, there's no reason to turn the system on at high levels. And in this case, when glucose is high, levels of cyclic AMP are low. And that means cyclic AMP is less likely to bind to the blue CAP molecule, which means that the complex doesn't bind very well near the promoter. And that means it's not recruiting RNA polymerase. So although RNA polymerase can bind inefficiently to the promoter under these circumstances, transcription will be at a very low rate, and at the very bottom of the screen it says little transcription. So again, high glucose, low cyclic AMP, no binding of CAP at the promoter, RNA polymerase can't bind well. So activity 3 asks you to consider two slightly more complex scenarios. What if neither lactose nor glucose is present? And then scenario B, what if both are present? And it's asking you three questions. Is the repressor bound to the operator? Is CAP bound to the operator? And overall, is lac operon on at high levels? And the best way to answer these questions is to draw some pictures. So on this slide, I've shown you my drawings. And again, you should try to make sure that you can do these drawings yourself before exam 2. First scenario, no lactose, no glucose. So because we have no lactose, that means our repressor molecule, encoded by LAC-I, can bind at the operator. Because there's no glucose, we have high levels of cyclic AMP, forming a complex with the CAP protein, which is recruiting RNA polymerase efficiently to the promoter, and that means transcription will happen. So is the repressor bound to the operator? Oops, yes. So that actually means that despite the fact that cyclic AMP and CAP are trying to recruit RNA polymerase here, the repressor is going to trump that situation. And because the repressor is bound, the lac operon will not be on at high levels. Okay, so I had to kind of contradict myself there, but that's because I wasn't considering both of the scenarios together. And that's what this question is trying to ask. So repressor is bound to the operator, CAP is bound to the promoter, but the net effect is the repressor is sort of more important. So lac operon is not on at high levels. What about scenario B, when both lactose and glucose are present? Well, when lactose is present, that means the repressor will be bound by lactose, and it will not bind at the operator site, which normally could allow it to turn on. However, glucose is present, which means there's only low levels of cyclic AMP, and it's not forming a complex with the CAP molecule, which means RNA polymerase is not recruited strongly to the promoter, and so we're not going to get strong transcriptional levels. So just to go through that again, is the repressor bound to the operator? No, which we might think would allow for high levels of transcription. However, the presence of glucose means that CAP is not bound at the promoter. And that means RNA polymerase can't be recruited strongly to the promoter. So we do not see the lac operon on at high levels, only low levels. And again, this kind of makes sense that it's not going to be on at high levels in either scenario. It makes sense that the lac operon doesn't need to be on when there's no lactose. And in the second scenario, it makes sense that it doesn't need to be on because glucose is present. So that is our wrap-up that we just talked about. Although these answers were different for repressor and CAP, the ultimate answer is no for both scenarios about whether lac operon is on at high levels. Our wrap-up of day 12 was our understanding of how gene expression is regulated in bacteria, and that is usually at the level of transcription. We learned about operons, which are usually or most commonly seen in bacteria. And we learned about the negative control of the lac operon, which is a repressor protein encoded by LAC-I that binds to the DNA to inhibit transcription. We learned about inducible operons, which the lac operon is an example of an inducible operon. The transcription is normally off, so as not to waste energy, and only when lactose is present is it going to be turned on. So we can say the lac operon is a negative inducible operon, and the presence of lactose inactivates the repressor to induce it. And then lastly, what I just talked about here is that the positive control of the lac operon occurs through catabolite repression with that CAP protein. High levels of glucose are going to result in low levels of transcription of the lac structural genes. I hope that helps with your work on Activity 3 and the Achieve homework.